The overall goals of the ARC study are to find uh, or develop biomarkers that track well with ALS progression, because that is sort of an unmet need in the field. Another one is to develop models that we can use in the lab, like animal models. And that would come from something like doing whole genome sequencing from participants. If we find maybe a genetic variant that was not previously studied, but is, related with maybe fast progressors and bulbar onset in our study, that might be interesting, an interesting variant to study. So we would want to test that in early cell models or animal models in our lab. So it's to help guide our decisions in the lab. And then overall, just to understand more about ALS and be able to determine maybe like subtypes of ALS or stratify patient populations. Because as we know, ALS is so heterogeneous. It's really hard to sort of categorize people into different types of ALS. We're not there, and that has large impacts on drug discovery and clinical trials, because we don't know if a certain drug might work on a certain population but not another. And we can't get to that answer unless we characterize ALS better than we do currently. So by collecting all the data we do on ARC, another aim is to get to that level of characterization where we can say, okay, maybe this therapeutic will help this group of people who have this presentation of ALS.